Neural mechanisms in aggression. Discuss the role of neural mechanisms in aggression, 16 marks. The limbic system. Deep within our brain is a network of systems called the limbic system. Biopsychologists have attempted to link this system to the emotional behaviors such as aggression. The key structure is the amygdala, which assesses and responds to threats in the environment. The more active your amygdala is, the more aggressive you become because they respond to threats and challenges in life. AO3. Research support for the amygdala's role in aggression comes from a study by Gosbeck et al. Researchers carried out fMRI brain scans on participants in a lab-based game called the Ultimatum game that provoked aggression. Scans showed aggressive reactions were associated with a fast and heightened response with the amygdala. Also, participants were then given benzodiazepine, which reduces arousal of the autonomic nervous system before the game, and this involved the number of aggressive reactions and decreased amygdala activity. This is strong empirical evidence for an association between reactive aggression and amygdala activity. However, a weakness of the study is that it lacks mundane realism. Results from people playing a game which involves many rewards cannot be generalized to everyday situations of aggression, such as starting a fight with someone. Therefore, this study cannot be extrapolated to real-life situations, and this reduces the internal validity of the research into neural mechanisms in aggression. Serotonin levels. Low levels of the neurotransmitter serotonin are associated with aggression. Serotonin in normal levels has calming inhibitory effects on the neural firing in the brain under normal circumstances. Serotonin works to inhibit firing of neurons in the amygdala. As a consequence to low serotonin, the amygdala detects a threatening situation. It becomes more active, causing the person to have reduced control, act on impulses, and making aggression more likely. AO3. Research support into monkeys looked at the impact of serotonin and aggressiveness. It was found that vervet monkeys fed on experimental diets of tryptophan, which increases serotonin in the brain, exhibited decreased levels of aggression. Monkeys who were fed diets that were low in tryptophan exhibited increased aggressive behavior. This suggests that the differences in aggression could be attributed to their serotonin levels. This increases the validity of neural mechanisms as an explanation for aggression. However, a methodological disadvantage of this research is that it used animals. Monkeys do not have the exact same brain functioning as humans. Human beings are far more socially, emotionally, and mentally complex. Thus, we cannot extrapolate animal research to explain human behavior. This decreases the internal validity of the research and its support for serotonin levels as an explanation for aggression.